And to kick us off, it's going to be Sarah Bruningk. And I've got a little bit of history from Sarah, but I did actually know about her work prior to inviting her to this for a couple of reasons. But one of them was because when she did her PhD in radiation physics at the ICR in London, uh, she worked with one of my old collaborators, a guy called Mark Chaplin, and Jibin Powell. So that kind of introduced her to the world of sort of mathematical oncology. And I guess she was intrigued enough to still keep a toe in that. And then she went and did a postdoc at ETH in machine learning and data science and with a healthcare sort of focus. So one of the things that Sarah's done is sort of integrate modeling, experiments, and machine learning, which I think is sort of somewhat of the part of the new generation of scientists that we're starting to see emerge from our field. So let's have a big welcome from Sarah. We're very excited to hear what she has to say.
All right. Thank you so much, Sandy. And thanks a lot for having me for this presentation. I was actually super excited for this invitation. And I'm really looking forward to talk to all of you about mechanistic learning, like the joint marriage of a lot of the things that we do here in mathematical oncology. And I think what is coming up more and more from the data and computer science side on the AI side. I will talk a lot about examples from radiation oncology and how we want to bring kind of these concepts here into play. And also, hopefully, you will all share that vision that I think it's a very good place to be, kind of to bring the best of the two worlds together here. So I wanted to start a little bit with the motivation. I think what we all do here, right? We all strive to improve cancer care for our patients. And one of the ways that we aim to do that is by personalization. So we want to move away from kind of this one size fits all approach towards accounting for all the rich information we have about our patients. We have molecular information, we have images, and we want to take this further to a tailored approach for the individual, where we want to really account for that treatment that works best for this particular patient. But this is actually not that easy, and there's actually a lot of different degrees of freedoms or knobs to turn on how we could approach this. And as I mentioned, I will be talking here about radiation oncology. So radiotherapy, we already had a very nice didactic from Heiko about the basics of this, so I'll not go into detail. But I wanted to bring back kind of the different ways on how we could personalize radiotherapy. And one way is about fractionation. So kind of the number of fractions that we give or how we kind of distribute the dose over time. So longitudinal aspects of the treatment. And this is really one thing I like about this community in particular, because there's always this dynamic aspect and how we can change things as they evolve dynamically. What I'm showing here is a paper that actually originated an IMO workshop, so I thought this was a good occasion to mention this. Long time ago, in 2019, Heiko was leading the team. We also won, I should mention, so no pressure. But this was an approach on intermittent treatment. So we wanted to investigate, is there potential for spacing fractions out really by long time for particular groups of recurrent cancer patients? If you're interested in this topic, please read the paper. I will not talk about it more than this, because I would like to focus in this presentation on a second degree of freedom, and that is the spatial dose distribution. As you may know, external beam radiotherapy, you have kind of the photons coming from different directions, aiming to concentrate the dose to the tumor. You inevitably always will strike the healthy tissue. So it is really an optimization problem. And I think it's one of the unique cases where there is already personalized therapy happening already today in the clinic, because each patient gets an individual treatment plan. So we calculate that for the individual. But it's not that easy. And actually, especially in cases like diffuse tumors, and we had a number of talks already talking about glioblastoma, I'll continue in this direction as well. We have to account for what is the target. And in radiotherapy, there is three types of targets. There's the visible tumor, then there is the invisible tumor, so that is the microscopic infiltration. We call that the clinical target volume, or CTV. And then there is a third one, which I will not discuss, but that's about the geometric uncertainty from the physics side. But how do we actually define something that we don't even see on the image? And this is where a lot of mathematical oncology projects also come in modeling cell distributions. But if you think about what is, or not think about, if you look what is actually done clinically, I would like to guide you to the 2025 EANO guidelines. This is defining how you should draw the clinical target volume based on what you see on the image. And it's actually an isotropic expansion, meaning we throw everything overboard that we know from a biological perspective of how tumors grow and where they infiltrate, and we just assume it's uniform. So I think in terms of approximations, that is a pretty big one. We do account for anatomical differences, so yes, we do not extend it beyond the skull or inside the ventricles, but it is a very crude concept. And I would like to discuss a little bit on options on how we potentially could adjust this, bringing also biology-informed growth mechanisms into clinical applications. And then there's the third degree of freedom, which is the actual dose. Yeah, so you can control the time, you can control where, and you can control how much. This is a very nice study that was presented last year at the SNO conference, where they actually used an AI model to predict the location of recurrence in glioblastoma patients and then boosted these particular regions. And I think it's really astonishing because they actually showed for the first time in blue is kind of this personally adopted or personally escalated is a better word, cohort, that there could be an improvement in overall survival for these patients. So with that in mind, how could we actually achieve that? Or what is it that we would like to investigate, right? We really want to go for the best approach for the patient. And now coming back a little bit to the data science perspective, how do we do that? Usually we have a predictive model that describes us how things are currently being done, right? So if I have a first step, I can at least make a prediction of how this particular patient is going to respond given this particular treatment and how we have always done it. However, the question is, what would it look like if, right? And from a computation perspective, this is a counterfactual analysis. I want to change aspects of my input features, or in a mathematical term, in kind of the way I deliver a treatment. So I want to extrapolate beyond what I have observed in my data. And I think this is a concept that is shared between these kinds of two worlds. And especially, I think, in terms of extrapolation, it's a clear advantage for mechanistic models. But I want to mention that we can also change inputs to AI algorithms. So what actually is AI? I mean, we will talk about this more today. I think we have a whole session on mechanistic learnings and combinations. But I thought it's good that we're all talking about the same thing here. And I thought who better to ask what AI is than the AI themselves. So this is a definition from ChatGPT. But what I don't like about it, and this is something I also get when I talk to my undergraduate medical students, so what is AI? And, you know, there's the word artificial and intelligence in there. And so there's often kind of a conception of there's something thinking and intelligence, which I think actually is not the case. So when someone asks me, I like to phrase it as it's a very complex optimization problem. So we're really trying to approximate a connection between our input data, the AI community calls these features, and our output, which is a certain label in a supervised setting. And I don't need to know about how this connection would come to be. And I think this is the biggest difference between kind of the data-driven approach, where we approximate this connection from examples, and our mechanistic approach, where we try to approximate our understanding in the form of equations. And as such, of course, the essence of the AI is the data. And then if you look, right, I mean, there's tremendous amounts of papers on AI, also AI in healthcare. And if we look and are honest, I mean, everyone here is using AI most likely in some capacity. And that is because it works extremely well in specific examples. Those examples where we have a lot of diverse data. But what is the reality if we go to oncology settings? this is a variety of data, right? So we have really rich information. We have information on the treatments. There's molecular genetic characterization. This is high dimensional, in-depth information that has to be processed. A lot of information actually is in text. I think this is very important to consider. And then, of course, there's images, a variety of different images ranging from MRIs, CTs, PETs, but also histopathology. So we can certainly not complain that we don't have data, but this data is what I would like to say real world, what is being done in the clinic. If we really strive to bring a model to application, acquired longitudinally. They are not all acquired at the same time, and there's usually a good reason for the specific sequence. There's also often a good reason for why a certain data point may be missing. So there's very often data missing not at random. And then you can tell me whatever, and maybe you are one of the very few lucky people and you have a huge data set. But for the majority of us, I would assume from the AI perspective, we are dealing here with very limited data sets. If you have hundreds, maybe thousands of patients, I think that is a good size. It is still small compared to the examples where purely data-driven models have really excelled. And in order to compensate for that, what do? We try to source data from multiple locations. This can be from within the same clinic and kind of more diverse groups of patients, or it can be multi-institutional, meaning that we inherently have to deal with the fact that data is not as beautiful and pretty as we have it in our predefined small data sets that we use for certain challenges. So in the essence, clinical real-world data is not very AI friendly. And how do we deal with that? So this is now where the mechanistic learning comes in. This slide is kind of the essence of contrasting the two worlds. If you really take it to the extreme, where we have the AI models as the universal function approximators, they really highly parameterized, that makes them really flexible, and driven by the data. And on the other hand, we have our mechanistic prior, so the understanding, where we likely need few but very specific types and examples of data to validate how and to what extent our assumptions are actually still usable in a given application. So yes, often these models are smaller, but that doesn't mean they are easier to bring to a real-world application. But they are driven by the knowledge. And so mechanistic learning kind of is then the marriage of the two. Why should we throw overboard all this understanding we already have? And at the same time, why shouldn't we be okay with approximating those parts where currently we cannot understand or explain in very specific detail every single thing with a data-driven approach? So it is really about bringing these two things together, the knowledge and an approximation, because maybe convince me otherwise, but I think no mechanistic model can really account for every eventuality and be as flexible as we would like it to be in the real world. So with this, I would like to kind of present some of the work that we published last year. So bear with me, yeah? So things have changed in the last year as well, so keep that in mind. But I think it was a great effort together with John Metzger, Alvaro Kuhn-Lucke from the University of Oslo, and also Paul Macklin, to kind of bring together what different approaches actually are there for this mechanistic learning, and how could we actually structure these into different concepts so that it's kind of easier to understand and also easier to identify what kind of approach should you want to pick for your particular application. And the first thing I would like to stress, picture, I mean, it's a very basic graphic, but the idea behind this is to think not in two buckets, but think as a space. It's a uniform or like continuous distribution. There is no end of just mechanistic and just data-driven. Anything we do usually lies somewhere in this higher dimensional space, and it's a continuous combination of how much of which do you want to combine. And so what we came up with in this review is a structure into 4 different buckets. Yeah, so I like structure. So that's why we thought it would be nice to have different ways of how we could characterize different flavors of mechanistic learning. And I will go now through all of these four and give a little bit of examples, and then we will actually move to some of the mechanistic learning that we do in my group currently. So talking about the first bucket, which is sequential combinations, and that just means to apply one of the methods after the other for kind of a shared goal. And as such, it's also the easiest way of how you could combine these. And if you think now you have two types of parameters, there's different ways. Yeah, so you can do one first and then the other and swap them around. So a very easy thing is to use your knowledge to drive the input to your through your data-driven model. Very simple. If you are predicting a certain outcome that is related to DNA inducing damaging therapies, you would want to use information that is related to this mechanism. So you select, for example, specific genes associated with DNA repair as an input to your machine learning algorithm. So it's a pre-selection of features, very basic. You can also make an architecture choice based on your understanding. Very simple. If you use images, you want to use convolutional networks. If you're using tabular data, there's different types of architectures that can deal with that. Topological data analysis, if you want to use information about connectivity, right? I mean, these are just natural choices. I'm just telling you about the things you've already been doing, and we're just structuring them here in a different way. But of course, you can also turn things around, right? So you can also engineer input features to a mechanistic model. So for example, cell proliferation and death rates can be extracted and then used in a specific context. Or you can also estimate model parameters from high-dimensional data. So for example, I have an omics characterization. I can estimate a specific growth rate of a tumor or of a cell line from these characteristics, and then put them into a model and make a prediction with this specific parameter as one of the features. Moving on, so this was kind of the easiest combination. Then there's also the type of parallel combinations. And we thought this is nicely defined as kind of alternatives for the same purpose. So you can do the same thing with either data-driven or mechanistic modeling. And a great example of these are neural ODEs. I'll not go in depth and explain the concept. If you want to learn about it, please have a look at this paper from Chen et al. From 2018. But effectively, you want to consider a neural network as a discretized ODE, and you can harness kind of the continuous function of an ODE in order to more effectively solve a neural network in this context. But that doesn't change anything about the goal of it, which was solving a specific type of training problem. Then you can also use neural networks as surrogate models. Very basic examples, you generate a large amount of synthetic training data with a highly complex agent-based model. And then you can train a neural network based on the synthetic data to approximate this complex model and have a very fast inference. Very basic example here. And there's, of course, a variety of different ways of how you could actually facilitate this. It's just about conceptualizing this. And I think especially the synthetic training data has been becoming more and more popular. Here? Here.
Affecting the internet
I am now it's back. But
Yeah.
it stopped sharing probably. Yeah. Online, if you don't see it, let us know. But in the interest of time, let's move on. Next combination, extrinsic combination. So this is now going into a different direction. Yeah. So here we're combining two things, but for different. So a common goal, but adding different aspects through the different approaches. And I'll explain you what I mean. I think a great example are digital twins. And we had a lot of sessions about this as well. There's different definitions of how you can define your digital twin. But let's be realistic, there can be different contributions. You can have a data-driven contribution to use unstructured data to make a specific prediction for that particular patient. And you can also have a mechanistic description to describe, for example, dynamical processes in this combination. Then there is also complementary post-processing. What I mean with that is we can use a mathematical analysis for interpretability to kind of shed some light into these black boxes. And the other way around, data-driven analysis of mathematical simulations as well. What is, of course, the essence of what I think everyone is much more interested are these more intrinsic combinations, when we really intertwine mechanistic understanding and data-driven. And I think one of the most popular frameworks in this context are physics-informed neural networks. I don't know who came up with this name. I think, well, because they actually used it in a physics context, right, for solving very key equations of physical laws. But it is, I think, a very nice concept because what we're trying to achieve here is to constrain the solution space to physically relevant solutions. And we achieve this by providing a second contribution to our loss function. As I mentioned, AI is nothing more than a complex optimization problem. So you're kind of minimizing the deviation between your labels and the predictions. And we add kind of a mechanistic loss contribution here to this concept as well, so that we can regularize our approach to these more meaningful subspaces of solutions. So what we achieve here effectively is we reduce the overall complexity of the problem, and therefore we can achieve a faster convergence in terms of given the same size of samples that we're using in this context. So pretty much you're just making it easier for your loss to be reduced. And I also want to highlight kind of a more recent expansion from Lena Podina, where they used a universal pin, so where you also approximate certain aspects of your equations, which may be unknown in terms of the functional terms. Yeah? So in the original one, you had to really define your PDE, but here you can also approximate aspects of that. And then in contrast to physics-informed, the biology-informed neural networks, and I think these two terms are often actually mixed, but we define it as when you constrain the architecture. So previously we constrained the loss, now we're constraining the architecture to known biological mechanisms. Take a gene regulatory network, right? So there's a lot of understanding that went into that and how and which genes and pathways and transcription factors interact with one another. And we can encode this in neural network architectures. We can encode this also very well in graphs. And so these are the biology-informed networks, where you really have a dedicated choice of architecture rather than kind of just fully connected neural networks. And then lastly, there's hierarchical approaches. Simply speaking, this is a sequential model, so mechanistic plus data-driven combined, but we train it end to end. And the advantage is that like this, we can actually also propagate uncertainty between kind of the individual level and to the population up about this. And there's especially work from Sebastian Benzekri, where they are applying this also in the oncology context for breast cancer and head and neck cancers, actually. So this was a fast track overview of what mechanistic learning is and how we can define it. So I hope now we're talking all roughly about the same things. I want to give you now an example from our group, which is a sequential combination, actually. So it's one of the easier types of models, aiming for spatiotemporal growth predictions of brain tumors. And this is work done by Daria Laszlo and Eftimis Georgios, a postdoc and PhD student in my lab. And I would like to introduce you to pediatric diffuse midline glioma. We've heard a lot about glioblastoma, so it's brain tumors. DMGs are also brain tumors. But let me put this into perspective. This is a brain tumor located in the brainstem, affecting predominantly young children with a peak age around 5 to 9 years of age, with a particularly poor overall survival. And this is because these tumors are located in one of the most vital parts of the brain, meaning that you cannot surgically resect them. And there's very few treatments that actually cross the blood-brain barrier to really make a difference for these particular patients. So radiotherapy really has an important role in the treatments of these brain tumors. On the plus side, this is a very rare type of tumor, but that doesn't mean that we can't use AI solutions in this context. It just means that, yes, here are all the problems I previously mentioned on small data, multi-institutional origins are just emphasized. But the fact that there is no surgery in this case also means I can observe dynamically a tumor as it responds to treatment with imaging. And I don't have this disruption of this whole system through a surgical intervention. So really here, imaging is the cornerstone, not only of the diagnosis, but also of the assessment. And for those of you who maybe have not worked in the clinic yet, but let me take away some kind of dreams that I was very surprised about. Clinically, we do not look only at the volume and the dynamics and all of that. Well, no, we just look at very specific aspects of the image. We look at different contrasts, yes, but then we reduce this complexity typically to biaxial measurements and assess kind of the difference in this product that is spent by these two axes relative to a baseline image. So a very, very basic comparison of response assessment, here specific to pediatric neuro-oncology. But I just want to emphasize the difference between what we observe in the research domain of our beautiful trajectories and what is actually done clinically. And very fresh from the press, I think it came out day before yesterday, there is now new guidelines from the AI RAPNO group on how we should incorporate AI solutions for automatic segmentation, but also what processes we need to put in place in order to bring this to clinical applications. So if you're interested, have a look at this. I think there's some really cool concepts, especially in the pediatric domain, where there's a much bigger push now for evaluating longitudinal context, considering also synthetic controls, so what a patient would have looked like in the absence of treatment, because we don't run large-scale randomized trials in these populations. But what does this actually mean? So let's assume for a second that we had an auto-segmentation model and we actually could provide longitudinal trajectories. No model is perfect. We've heard this in a variety of combinations already. And we are very much aware that segmentation is difficult. We can quantify this in different ways, and we will get slightly different results. But what I wanted to show with this is 3 state-of-the-art models, and in red is the manual segmentation, is that, yes, the overall trajectories, the absolute volume is different, but the trends remain the same. And I think the trends is really what we as the kind of community that is much more in longitudinal processes are much more is most relevant to us. And I also wanted to show these are 2 patients with very different trajectories, even though they received exactly the same treatment. And this concept here of pseudoprogression really is something very important in the domain of radiotherapy for brain tumors. And so the question is, how can we account for all of that, keeping in mind that clinically, we're used to looking at these images, baseline versus current image, and it's really not looking at a trajectory. So what that means is, shouldn't we provide predictions in the space that is most used by the clinicians? And in this case, this is the radiologist, and that means that is an image. And that brings me back to my dogs that I showed you initially on this personalized therapy slide. And if you were wondering how I generated these images, this was using a so-called guided denoising diffusion vision transformer based models here in ChatGPT. But the concept is pretty much that you go from a noisy version with a cue, and that could be text-based, but there could be other ways to guide these to provide an image from a noisy version of itself. And what we did in this particular study is actually to use the same concept to abstract kind of how brain tumors grow. So we could generate a brain tumor image from a noisy, very noisy version of itself and guide it towards different characteristics, for example, a different size. And so the aim of this project was not really, we did not come up with the denoising diffusion models, right? So other people have come up with this, but we wanted to see how realistic are the images that we can create with this. Do they actually recapitulate tumor growth? So whatever the larger tumor is, does this actually match the way the tumor grows in a particular patient? And also, can we translate this to probability, so likelihood of in which anatomical region a tumor is most likely to progress through? So we used a bunch of data in this project, and I want to emphasize the data that was used to train this model is not longitudinal. So we actually used a public data set, the BRATS challenge data comprising over 1,200 brain GBM cases, to first of all learn how brain tumor images and brains look like. And then we refined this on a small set of around 140 pediatric patients. It's multi-parametric MRIs. And I also show you here these histograms because they come from slightly different anatomical locations, these two types of tumors. So one is hemispheric, the other one is in the brainstem. And then we have here, for this purpose of this presentation, it's a small validation set, 13 patients with longitudinal information. So there we really have information on how the tumor actually grows. And this, of course, is what we would like to compare in the end. So how does this work? Yeah, I will not go in depth, but the concept I think is actually quite nice. First of all, you add noise to your image. There's nothing to learn about this. We just add this. Yeah, so stepwise until we have a very noisy version of this image, preserving just a subset of kind of main characteristic features so that we can obtain kind of a reconstruction still of this particular brain. And then what the model actually learns is this denoising process. So first of all, we train to just reconstruct the same version of the image from its noisy self. But what is nice, as I mentioned, is that we can guide this process. And in this particular case, we guide it with the gradient of a segmentation model that estimates the size of the tumor. So we continue to guide it towards an output saying like, I want this tumor, but at this predefined size. And what happens is that I get the same brain with the same type of tumor, just at a different size. So what I showed you here and what we train actually is a two-dimensional model. Yeah? So these are intense to train. At the time when we started this project 2 years ago, actually GPUs were not large enough, at least in our facility, that we could have effectively trained this in 3 dimensions. So we trained it slice-based, but then also wanted to see, can we actually stack back to obtain the full tumor? And here we harness kind of 2 aspects of this. We wanted to have the stacking. We also wanted to have the probability. And the nice thing is actually that this is a stochastic process. So if I renoise this image, it's slightly different noise level, I will obtain a slightly different output. So if I repeat this process, I will obtain a probability map of which pixel in the final larger size are tumor, given, let's say, 100 iterations of reconstructions. And so what was then nice to see is that if we stack these probability maps, we actually get a smooth distribution of our tumor, so we can reconstruct a 3-dimensional area, 3-dimensional volume in this case. So how do we quantify the performance? I was mentioning this is kind of the essence, right? You want to see how realistic is this. So first thing we do is we do what the AI group do. We calculate a bunch of metrics. I'm not sure if anyone here knows what structural similarity is or this FID metric. Let me tell you, to summarize, it was pretty good in terms of these metrics. Yeah? So the structural similarity should be close to 1. We compared the performance in adult and the pediatric subset. We also calculated the peak signal to noise ratio, and then this FID metric, which should be as low as possible. But what does this tell us about how realistic these images are? Because these metrics actually were designed for real photos. Yeah? So they were not designed for MRIs, and also designed for really large-scale data, and especially this FID metric is very sensitive to the size of your distribution that you're looking at. So we turned to our human observers. We did a study in n equals 5, and I thought we would do a brief study here, and I hope this will work now. Yeah, in n equals, I don't know how many we are, like 30 to 40. So I would like to ask you, who thinks there is a generated image in here? And if so, which side of it is it? Is it the right side or is it the left side? So I would say we take a vote. Maybe we take a vote. If you think it is the right image, which is kind of this one closer to me, yeah, raise your hand. Being real, yeah, right image real. Okay, just to confirm, left image real. So in terms of probabilities, I think there was a lot of non-raised hands. Yeah, it seems a little bit difficult. Let me give you the answer. So the right image here was actually generated and the left one was the real. Okay, first try, right? So I think it was maybe difficult. Have a close look again. Maybe you can see what it is. And let's try this one more time. So is it the right image which is real, or is it the left image which is real in this case? And I would suggest we take another vote. So who says it's the right image which is real? Okay, so there's quite a lot. Just to check, left image which is real. Okay, so we seem to have a slightly larger proportion here for the right one, which in this case actually was correct. Yeah, so the right image here is the generated ones. But what I wanted to show you here, I mean, it's also a screen, it's a small image, it's hard to see. Don't be fooled by the pixel. Yeah, this does not mean it is generated. But it was a difficult task, right? And actually, if we asked some experts in this domain, we had 3 clinically board-certified neuroradiologists, and we had some clinical fellows whose job it was mostly to contour brain tumors. They also were unable to do this reliably. And we quantified this in a variety of different metrics. I mean, they were a little bit better maybe than random, similar to what we had here, but there was you could not say they could actually distinguish these types of images. And this was a large set of images that we evaluated. But what I want to draw your attention to is these guys, the blue ones. So this actually was a UNet classifier that we trained for the same task. And yes, the performance also isn't great, so our images are fairly good, but it's also not as close to random. So there are still underlying aspects in these images, which mean they do differ between real and generated. And we looked more deeply, and actually it's relating to features of the underlying image noise, which in hindsight makes a lot of sense, right? It's a denoising model that we're using here. So that was interesting. They look pretty real. We also went a step further, and in the interest of time, I will go a little bit quick through this part. But we also wanted to see, what are they actually looking for, right? So if I now were to ask you guys, whoever voted correctly, what is it that you were looking out for in these images? What are the most important parts? And this was a study together with the University of Applied Sciences in northwestern Switzerland, where we had 13 medical experts and 21 AI experts, because I think this is complementary expertise, to do the same task. Actually, they had a comparison of different images, either just one image shown or a comparison one versus the other, to judge which one is real and which one is fake. And we used eye tracking to check which parts of the images were they actually looking at. And the results were actually quite nice for this first. Again, if you were just showing one image, they also were unable to identify that. However, it was interesting when we went to the second task where it was paired. So they saw 2 images of the same tumor. One was real, one was fake. Some very smart people were actually able to identify this with more than random performance. It was never perfect, but there were a few people that actually could make out what this was. And then what we looked at is actually then differences in terms of the attention patterns from this eye tracking. And there was a clear difference in the sense that medical experts always looked very specifically to the tumor. AI people looked much more general overall. But it was also very hard to make out why were some of these people actually able to identify the differences here. And this is something we're still looking into now. But let's move on. So what about the longitudinal aspect, right? So I'm here to talk about dynamics. So is this now also realistic? They look good, but is it how the tumor actually grows? And for this, we used our set of 13 patients. We went to a slide-based level, so 178 slices, and quantified the agreement between the real tumor extent and the generated one from these probability maps. And we quantified this here in terms of dice. I want to stress, I mean, dice has a lot of issues, yeah? So I think this is something we need to consider in this context. But what I would like you to look at is actually this example here. The original tumor was actually in this location and then kind of expanded to this region over here. And the model really nicely picked that up. So it was really about identifying also the directionality of that growth, which was a very good aspect and really what we were after in this context. I also wanted to show here that there are still some problems if you extrapolate too far. So if we increase our volume by 200%, and this can happen for very small tumors, we see that we have kind of a discrepancy in terms of the true obtained size and the generated one. And that comes down to the point for what we trained this model. This model was trained in the range of 25% increase. That's what clinically is considered relevant. And so just a caveat to keep this in mind where you apply this. But what about the time, right? So all of what I just showed you is generating tumors of a predefined size. But unfortunately, I don't know the size, right? So we want to know how does it grow. But now I give the word back to all of you guys, right? This is what we do a lot in mechanistic modeling. We look at trajectories, we fit them, and we make predictions of what the volume of a tumor or particular marker is going to be at an extrapolated time point. And this is what we did here as well. So we used, and in the spirit of the, what are we calling it now? The Enderling razor. We wanted to use a very simple approach to describe this because we have very few time points for this patient. So we used an exponential tumor growth. Keep in mind, we're using this model only for a very small interval. We know tumors don't grow exponentially. This is an approximation in this context for this limited amount of time. One compartment. Then we have our form of treatment, radiation. We separate into two compartments based on a given surviving fraction. So that's the next parameter. And then we have a changed growth rate in this dying compartment, lambda star. But lambda star, actually, we wanted to be kind of flexible. So this is where we burned all of our parameters. I wanted this model to be flexible to account for differences in cell death mechanism because we know different drugs introduce different things in the cells, and that introduces a whole cascade that affects how this tumor dynamically is going to respond to this treatment. As we saw in these two tumor growth trajectories, some tumors have this progression initially and then they will shrink. And especially for radiation, I think it's important not to model this as an instantaneous event, but to account for kind of the delayed cell death that is introduced here. So we used a sigmoid function for this. And I want to stress at this point, I'm not a mathematician. I did not look at the identifiability of this approach. Very happy to do that if someone is interested. But we wanted this to be flexible in terms of delaying it and the extent of how quickly this tumor is going to shrink. And shrinkage would happen then at the same rate as it was growing initially. And now I want to also motivate where this model came from. It did not originally come from patients. Actually, this model came from a spheroid experiment that we did together with this company. They have a really cool robotic system where you can add drugs, you can wash them out, you can image the spheroids, and you can do this in high throughput over long periods of time. So excellent types of data. We did this for diffuse midline glioma spheroids. And actually, this type of model was flexible enough to describe these differences in dynamics that we I think we had 10 different types of cell lines. and 20 different drugs that we tested, and also at different concentrations. So it was kind of flexible enough to describe this. And now we took this same approach, very basic, and applied exactly the same framework to our patient data. And this is what comes out of it. And if you're interested, have a look at this QR code. This was just recently presented at MICAI in a workshop on longitudinal modeling as a paper, where we actually now calculate the tumor size from this external mechanistic model and then guide our image generation towards the size and in particular the probability distribution that is defined by this mechanistic model. So what does this look like? First of all, let's have a look. Does this also actually work for the tumor growth trajectory? So I'm showing you here kind of one fit using also a Monte Carlo approach to account for the uncertainty in these data. And then the same kind of data, leaving out the last data point, which is what we're interested in. And you can see, of course, it's not going to be perfect. We're still kind of covering the data point in this particular case with our confidence interval. 95%. But if we look at the overall performance for 9 different patients, and we quantified this as R square and also as normalized RMSE, we actually see that this is quite variable. Yeah? So for some patients, this looked pretty nice. For some patients, I mean, this R square really was looking to an extent where I would potentially be worried. But at the same time, I want to remind you, this was these volumetric curves. We have a lot of uncertainty in these curves. And we thought we will just go ahead, we will still use this model because we have also a stochastic process in the image generation. So pretty much we're bringing here together 2 types of processes that we're sampling from, the size and the image generation. And so then we applied this in this context. So we leave out this last data point, predict the size, resample, obtain our growth probability maps, and then we compared this. And here I'm showing you an example that was guided towards the true size, for which we previously showed that those images actually recapitulate the growth, and then guided towards the estimated size. So yes, there are differences in the probability distributions, but the anatomical extent of them, they still agree. And if you quantify this with a Hausdorff distance, observed that we were very close to what we would have achieved with the true size of a particular image by guiding with this mechanistic approach. So coming to an end, how would we know the parameters, right? Because let's be realistic here, a lot of my data points I cannot yet predict here. Yeah, so it would be really nice if I could predict my growth rate or my surviving fraction before going into this. And there's a variety of ways of how we are looking into doing that. And unfortunately, I'm not at a position to say, oh, here is the solution. It is solved. It works like this. But I do want to point out 2 projects that we're currently working on. One is a project with Lawrence, where we're using multi-omics data to predict radiosensitivity. So the S, alphas, betas, using self-supervised learning. So this is not a mechanistic learning per se. We're using some biology-informed networks here, but it's really going towards a prediction of this parameter. And then secondly, this is a project together with Julia. This is about using spatial information obtained from histopathology images. So here we have an AI model that predicts phenotypes of cells from H&Es to provide kind of spatial insight from the data modality that we have available at scale, which is H&Es. This is heavily based on digital pathology foundation models, so models really pre-trained on large amounts of data. And then we validate this between whole slide images that we and that was really what Julia did a lot. We have an H&E, and then we also stained it for a particular protein marker. Here I'm showing you histone 3 K27, and this is a mutation that is particular to diffuse midline glioma. So this specifically means distribution of tumor cells in these samples, and then comparing the predicted distribution of H3K27M mutant cells to the true distribution obtained with immunohistochemistry, in this case, immunofluorescence staining. And that actually also works to some extent already. So I would like to thank you for your attention. I would like to hope that you are motivated to dive into aspects of data-driven approaches, combine them with mechanistic learning for a variety of different tasks. Of course, this was not just work done by me alone, but there's a whole team behind this, and I really want to thank them. And I want to thank, of course, also Sandy and Kirsten for inviting me here today. And I will end with this slide. If you are interested, we are looking for PhD positions at this point as well. Thank you.
Okay, thanks, Sarah, for a lovely presentation. Very clear. There must be questions.
Sarah, beautiful. So you set out with trying to improve dose painting.
Yes.
Which I agree is an incredibly important problem. So we haven't seen any about this in your talk, though.
No.
So would
Yes.
you be able to use some of kind of these diffusion models to see how will the tumor evolve during radiation to see where residual pockets would be to dose escalate prospectively? How much data would you need to be able to do that?
So the first thing is I want to emphasize what we showed here actually is a CTV definition already. So you have a map of where tumors is most likely going to progress through. So that was speaking kind of to the definition part. We are actually now looking into the dose aspects of things, and I think this is actually super interesting. I think we will need not that much more data. So I think the beauty of these types of diffusion models is, as I showed, we will train these in a different way. Here we will have a separate approach that will describe the response of specific aspects to dose, and we have a three-dimensional dose distribution. We can have that in slices as well. And then we will have an outcome given an image and given a separate diffusion model for the dose distribution in this context, which are linked together as well. So this is something we're currently looking into. We don't have results on this yet, but yes, this is what is coming up next. And then, of course, the motivation there is then you have a tool for counterfactual building, because then you can change your inputs, you can change the dose distribution that goes in and see what does it mean in terms of image of what comes out of that. Yeah.
Beautiful.
Thank you. That was a really cool presentation. I was wondering, you mentioned the power of pins, and then have you considered with your diffusion model, rather than having it guided by just the size, have it guided by, say, a reaction diffusion equation, which we know work quite well in brain tumors?
So this is we're currently working on. So I was thinking, should I put in things that we currently do and then or not? So I didn't choose to put this in, but yes. So I actually had one slide. There is physics-guided diffusion models. And yes, we have a very good description of how tumors could be approximated to grow with DPI kind of models, or this Fisher-Kolmogorov type equations. And this is what we're looking into now. I also should stress this approach, I mean, we started this 2 years ago. It's in pixel space. Now we work with latent diffusion models, which are computationally much more efficient. And therefore, we'll also have kind of this physics-guided latent diffusion to arrive at hopefully more efficient training regimes as well. Yeah.
Cool. Thank you.
I saw a question over there, I think. Yeah.
Thanks for a wonderful talk. I have a broader question about scientific machine learning. So we've kind of gone from feature engineering to loss engineering to regularizations, learning operators, and governing equations. And now we are, flow matching, and we're doing building multi-agenic systems. So I'm curious on your thoughts on whether we should maybe consider moving from biology-informed neural networks and kind of mechanistic learning more towards biology-informed context, because we are doing more context engineering these days.
But where do you draw the line, right? I think to me, this is a flavor of this subset. It is about where do you want, because the context to me is also understanding and knowledge, right? So you would also infuse this, and I think this would be very relevant at this point. At the same time, I also think I would be wrong to recommend anyone here what they should do. I think there is, what I wanted to show here is like there is different ways of how you can approach things, and you should pick that type of approach that works best in your particular scenario, and that defines the question you want to answer, and that is defined data you have available. And if there is large contextual information on your specific problem, yes, you should harness that. If there isn't, but you have a ton of longitudinal trajectories for some reason, I'm not sure this would exist, but then maybe the answer would be different. So I think mechanistic learning is something that I would consider coming up and running. It's a very, very broad topic, though, that I think is not very clearly defined where you would draw a line in this context. I do want to stress, though, and I was thinking of putting this here as well, but agent-based models and how we incorporate knowledge and knowledge graphs in these types of approaches is, I think, something that is highly relevant today and a field that is very growing in kind of the AI domain currently as well. Yeah.
Any other questions? Oh, Masood. Well, while I'm walking down there, I just want to follow up on his question. So are you saying that people are using machine learning approaches to train agent-based models or to set the rules? Because to me, that's where the biology should come in.
Okay. So there's two types of agent-based models. Yeah? So there is the agent-based models, let's say, on how we understand them with the specific rules, and we control kind of learning emerging properties of a system from the individual agents. And then there is the AI definition of an agent-based models, where you have different LLM-based approaches that are given a specific task. You can have one AI model that is trained to check the quality of a specific segmentation. You have a second agent that generates that segmentation. And then you have a third one which comes in and says, actually, you have to account for the overall connectivity, just an example. But there's different AI models, and we call those also agents. So it's kind of two different things in this context. Yeah.
I have a couple of questions. The first one is, as you said, using sections, different sections in memory image to make 3D projection or something. So how far are the two cross sections? I mean, two sections are.
So they are all resampled to 1 millimeter isotropic pixel size, but of course depends on your scanner what the original slice thickness was.
So how do you deal with something that might be missing within this between this 1 millimeter?
Within this 1 millimeter?
Yeah.
We don't. Our reconstruction pixel size is also 1 millimeter.
I see.
So then you have to, yeah, I think interpret. This is the essence of also an image, right? You don't know what was in between. This is kind of an agglomeration of everything that is in this 1 millimeter. Okay. This is the resolution, and yeah.
The second question is physics-informed neural network and biology-informed neural network. Do we say that physics-informed neural network is a subset of biology-informed neural network, or it's the otherwise?
Physics-informed is, sorry?
Physics-informed neural network and biology-informed.
Yes.
Which one is subset of the other?
Which one is a subset of the other? I think it's two different things, right? Or at least this is how we try to define it. Yeah. To me, I say the physics-informed is a construction, is a regularization of the loss. Mm-hmm. Biology-informed is a regularization of the architecture from the get-go. Yeah, so you already start with an architecture that is recapitulating your understanding of that biological network in itself.
So, the structure of the neural network. Architecture of the neural network.
This could be, yeah, it could be a neural network, it could be a graph neural network. I think this could be done in a variety of different architectures. So it's just to kind of constrain your connectivity in this sense. But you could also do physics-informed, as I said, on diffusion models, on neural networks, on convolutional networks. So this is a regularization aspect of the training then.
Okay, let's thank Sarah again for a lovely presentation.